Okay, sick. Okay, if anyone, so we should be live. Guys, if you can hear me, put a W in the chat. If you can hear me, put a W in, actually, there are people already pressing W, so let's use a different letter. If you can hear me, put a Z in the chat. Okay, no, it's fine. Everyone's putting W. Okay, perfect. Good stuff. Welcome back, guys, for round 2 of science. So we've already finished all of our paper 1, biology, chemistry, physics. I hope it went well. I hope that you guys got the marks you were aiming for. If you didn't, don't worry about it, because this is your chance to make up for what you think you lost on. Okay, so we're starting with biology paper 2 today. If it's your first time joining us today, this is we are ExamQA. We go through science topics the night before your exam, and we're going to try and cover as much as you need to know before tomorrow's biology exam. So we've got the admins in the back. We have changed our scenery a little bit. For those of you who've noticed, we're in a different location today as a one-off. Maybe there might be a surprise for you at the end of the stream. Maybe not. I don't know. Let's see. But my job here is to make sure that tomorrow's biology paper goes well. If you haven't revised biology all year round, don't worry. Here is where that changes. And if you have, still, you can join us. Let's see how much you know. Test yourself against everything we're going to cover today and see if you can answer all the questions properly. Okay. Now, we're just waiting for the green light from Zishan. Obviously, we're going to start in about 2 minutes. So, guys, make sure that you subscribe to ExamQA. If you haven't, we're very close to hitting 100k subscribers, and we'll get that nice little plaque afterwards. Obviously, I don't know what we're going to do with the plaque, but once it's there, we'll find out. And if you've got a friend that's not good at biology, then send this live stream to them, because you will save their life. Okay, now, guys. What's that noise? Is that the police? Ah, the police are here. Okay, guys, is that foreshadowing for something that's gonna happen today? I don't know. Maybe not. There's a bunch of weird stuff that happens in these live streams. I don't know what's gonna go on. Okay, but shout out to a couple of my students did ask me for shout out, so let's get through them quickly. So shout out to Alicia from Barking. Shout out to Zain from Barking, and of course, guys, if you go to Barking tuition and you get taught by Miss Hira, you are one of the lucky ones. Guys, you know what? I'm thinking if we get 5k likes, next live stream for chemistry, Miss Hira will teach you. Miss Hira is going to teach you the chemistry live stream. 5k likes, we'll do that. All right, so Zishan, are you ready to go? We're not ready to go. Okay, cool. So guys, out of biology, chemistry, and physics, type in the chat which one's your favorite. Is it bio, is it chem, or is it physics? Out of the 3, which one do you like the most? Because if you said biology, then test yourself. But if you didn't say biology, then here's where that changes. I think tomorrow's paper is going to be a nice one. Obviously, I'm not going to say that I've seen the paper. Maybe I have, maybe I haven't. But let's go through. We're going to go through some topics tonight to make sure that you get all of those amazing marks. And how was maths, by the way? Did you know the maths live stream? Did that help you? If it did help you, put W Zishan in the chat, so that way he can do the maths paper free. I think otherwise he's not going to do it. Cool. All right. So we're going to start in about 1 minute. Just waiting for the time to hit 34. Moderators, is everything okay at the back? Can we start?
No.
Okay, obviously you know our moderators are a bit bipolar, so today they're feeling well, but maybe mid live stream they might go a bit crazy. All right, cool. Okay, so 30 seconds before we start, guys. Don't worry about writing down everything. Biology is one of those topics where we have a lot of content to go through, okay? So don't worry about writing anything. Anything important, I'll tell you. If it's not important, just listen and put it into your brain for your exam tomorrow. And here we go. Guys, if the live chat is distracting you, if there's too much brain rot in the live chat, my suggestion is to just completely ignore it. Close the live chat and lock in for this one. Here we go. Here's your final biology lesson of your life, unless you do A-level biology. Otherwise, for most of you guys, this is going to be your final biology lesson ever. After this, no more biology. Zishan, ready to go?
Yeah.
All right, let's have Zishan on camera real quick. Zishan,
Guys,
grace us with your
Biology paper 2 tomorrow. Everything is ready now. The moderator room sorted out today. No problems today. No fights gonna happen.
Are you sure, Zishan? No fights?
I've got them on lock.
What about the ninjas? Any ninjas?
The ninjas, I've got them just to make sure.
What about the dinosaurs? Do you have any dinosaurs?
I hope not. Otherwise, I don't know what's going
to happen. We're going to have to slice them up.
That stuff.
Okay.
But this is your final paper 2s and 3s.
Yeah.
We are going to try our best to get you guys grade 7, 8s and 9s. If you subscribe, you're going to get notifications of when we do each of these.
Guys, Zishan, they're saying, what about the flamingo versus dinosaur fight? Is that even a real thing? What is this?
I don't remember any of them. I got kidnapped and they did the Men in Black stuff for me. All my memories are wiped when I got in.
They neuralized you, yeah? Talking about neuralized, we're going to go through that in a second.
Very good.
Alright then.
MPK fertilizers.
Yeah. Alright, here we go. Guys, lock in. Time to lock in. No more jokes, yeah? Here we go. Biology, paper 2 live stream. Let's go. Alright, obviously. First topic is the endocrine system. Why? Because it's such a large topic and there are so many hormones that you have to know. Let's go through it, okay? So, first of all, we know that messages are sent in our body in 2 forms. We have through electrical impulses, okay, and that's through the nervous system. So our nervous system communicates with our different parts of the body, and the nervous system is the brain, spinal cord, and neurons, okay? And then we also have another way that our body communicates with itself, and that is through chemical messengers, and that is part of the endocrine system, okay? So we have electrical impulses and we have chemical messengers that communicate with our body. All right, so let's quickly skip this part's not important. All right, here we go. So if in your exam they ask you what is a hormone, okay? A hormone, keyword here, is a chemical messenger that is produced by a gland. Now if you don't say the word endocrine gland, you might lose a mark. So hormones are chemical messengers produced by endocrine glands, okay? But where do they go? Where do they just fly around the body, or do they go in a specific place? So part 2 is they travel in the blood, and finally, and everyone forgets to say this, so make sure you don't forget tomorrow, they bind to receptors on a target cell. So that's a whole 2-sentence mouthful. Let's go back one more time. Hormones are chemical messengers produced by endocrine glands. They travel in the blood and they bind to receptors on a target cell, okay? And by that, they deliver their message. Now, when we said endocrine glands, we don't just have one, we have multiple in our body. So let's talk about the main endocrine glands that we have in the body, okay? So the first one, the king of all the glands, or the master, the pituitary gland, okay? That's the master gland. Why is it called the master gland? Because it activates the other glands in the body to make them produce hormones, okay? So all the other glands are waiting for the pituitary gland to tell them, make the hormone, okay? Now, the next gland that we have to talk about is the thyroid gland, okay? And that's right over here in your neck. Now, what does that gland do? Well, that produces a hormone called thyroxine, and that controls your metabolic rate, okay? So basically, your metabolic rate is the rate of chemical reactions in the body. And obviously, if your metabolic rate is high, then that means your body is able to metabolize things quicker. And if it's low, then it means it takes longer for your body to metabolize stuff, all right? But here's the thing, some people have a very high metabolic rate and some people have a very low metabolic rate, okay? And that's all to do with the level of thyroxine in their body, which can go up and down. And we have to explain how that works. So here we go. Here's how thyroxine levels are controlled by negative feedback, all right? So, number 1, pituitary gland releases TSH, which is called thyroid stimulating hormone, okay? So right here, the pituitary gland releases that. Where does that go to? That travels all the way down to the pituitary gland. Once it gets there, it tells the pituitary gland to produce thyroxine, okay? So TSH stimulates the thyroid gland to produce thyroxine. And as a result, what happens to our thyroxine levels? It starts to increase, okay? So we get more and more thyroxine, which is good. Your metabolic rate starts to increase. But what happens? How does the body prevent it from going over the top? Well, too much thyroxine, okay, too much thyroxine goes back to the pituitary gland and it inhibits it. So what does that mean? It means it goes back and says no more TSH, okay? So because it goes back to the pituitary gland and says no more TSH, what happens to the level of TSH? Well, the level of TSH begins to fall, okay? TSH levels begin to fall, and then because there's no more TSH, there's also going to be lower levels of thyroxine. Okay, everyone? So, one more time. Pituitary gland produces TSH. TSH goes to the thyroid gland, which makes thyroxine. When there's too much thyroxine, the excess thyroxine goes to the pituitary gland and says stop making TSH, and that causes the thyroxine levels to go back to normal. And all of this is called negative feedback. Alright, next one. The adrenal gland. You have two of them and they sit right here in your back above your kidneys. Okay, so not the kidneys itself, but they sit above your kidneys, and there's two of them. So what hormone do they produce? They produce the hormone adrenaline, and that's responsible for the fight or flight response. Okay, so when you're under, like for example, when you're suddenly stimulated, your body releases adrenaline. Okay, so what does adrenaline do? Two things. Number one, it increases your heart rate. So your heart rate goes up. Imagine before a roller coaster when you're scared, or before your exam when you're scared, your heart rate goes up, and that's caused by adrenaline. Yeah. And number two, it also increases the rate of respiration. Why? Because you want your cells to make more energy in case your body has to prepare itself for action. So those are the two things that adrenaline does. Okay. Number three, we have testes and ovaries, which obviously control sexual development. So testes for male and ovaries for female. And the hormone is testosterone and estrogen. Easy. Moving on. Okay, here's the big one. This could be where you get your 4 marker. So let's look in for this one, guys. Here we go. Pancreas. Now the pancreas makes 2 types of hormones, insulin and glucagon. And both of these hormones are related to controlling how much glucose is in your blood. Okay, so without these hormones, the amount of glucose in your blood will not be balanced properly. So let's talk about how this works. So let's start with insulin. Okay, so step 1. The question could be, explain how insulin maintains blood glucose levels after a meal. That would be the question. It's 4 marks. Let's talk about it. Here we go. Number 1. After you've had a meal, we know that there's a lot of glucose in the blood. Okay, so look, all this glucose in the blood, which is good, but we don't want too much glucose. Okay, so what's going to happen? Well, the pancreas will release insulin into the blood to help lower the glucose levels. So pancreas releases the hormone insulin. Because it's a hormone, it has to also go into the blood, just like the glucose. Okay, then the pancreas travels to another organ called the liver, and it tells the liver to absorb all of the glucose from the blood and to store it inside the liver. Okay, when it gets stored inside the liver, the glucose compacts itself and it forms something called glycogen. Okay, so you have 3 G words: glucose, which turns into glycogen. Yeah, and don't confuse these two with the hormone glucagon, which is the other G word. We're going to talk about that in a sec. Yeah, all right, good. And as a result, the concentration of glucose returns back to normal. Okay, so those were the 6 points about how insulin works. But what if the question flips it around and says, oh, forget insulin, explain how glucagon works? Okay, so let's talk about this. Now, let's say you haven't had a meal all day, you're hungry, or maybe you've just done some intense exercise. All of the glucose in your body, in your blood, has been used up. Okay, so now the blood glucose levels in your blood are very, very low. So if someone has by the way, side point, yeah, if someone has low blood glucose levels, one of the symptoms is that they'll be super tired, they'll be dizzy, almost like as if they're drunk, yeah, which is not good. Yeah, all right, so anyway, how does the body prevent that from happening? Well, the pancreas releases a different hormone this time. It releases the hormone glucagon, which also travels into the blood. Just like insulin, they're hormones, they have to go to the blood. Then it travels from the blood to the liver. Okay, when it gets to the liver, it tells the liver to break down the glycogen that was previously stored and release it back into the blood in the form of glucose. There you go. Okay, so glycogen goes to the liver, tells the liver to break down the glycogen. So glucagon, my bad. Glucagon goes to the liver, tells the liver to break down the glycogen into glucose and return it to the blood, so that the blood glucose levels can rise a little bit until it's back to normal. Okay, everyone? There you go. Now, let's talk about if our body is not able to control blood glucose levels, if we have uncontrolled sugar levels, this can lead to a disease called diabetes. Now, I'm sure you guys have heard of it, but let's quickly look at both types in detail. All right, here we go. First one, type 1 diabetes is caused by someone that has a faulty pancreas. So the pancreas, which remember the pancreas makes insulin, right? In type 1 diabetes, their pancreas does not make insulin at all. So we have all this glucose in the blood and we need insulin, but the pancreas says, sorry, I can't make insulin. I don't have the ability to make it. Okay, so people are usually born with this. So type 1 is caused by faulty alleles and is present from childhood. So if you have like a young friend who's injecting themselves with insulin, that's because they have type 1 diabetes. They're usually born with it. Okay, so how do you treat someone with type 1 diabetes? You literally have to give them insulin injections throughout the day for the rest of their life. Okay, that's type 1 diabetes. Now, type 2 diabetes. Okay, so just to quickly summarize, type 1, the pancreas cannot make insulin. Yeah, it cannot make insulin. As a result, blood glucose levels go high and high and high, and that's because of their faulty genes. They can't make insulin, and to treat it, you have to inject them with insulin. Good. Let's move on to type 2. Okay. Now, type 2 diabetes, the pancreas is completely fine. Pancreas can make insulin. But the problem now is with the liver. So in type 2 diabetes, the liver does not respond to insulin. Even though the pancreas is sending insulin to the liver is like, no, I've had enough, it's not responding. Okay? So insulin says to the liver, what? Absorb the glucose, put it into yourself in the form of glycogen, but the liver can't. It just can't do it anymore. Okay? The reason behind that is not because of genes, it's because of obesity. So this is more of something that the person caused to themselves because of an unhealthy lifestyle. Okay, so when someone has obesity, then the liver does not respond to insulin. So if you were a doctor, how would you treat someone that has type 2 diabetes? You give them one advice, which is exercise. Why? Because when you exercise, what happens to all the glucose in your blood? It will burn the glucose through respiration. Okay, so it begins to drop. But if that doesn't help, then we can give them insulin injections. But first, they have to exercise, and also they have to change their diet so they don't have too much sugar, okay? And then if that doesn't work, you give them insulin injections. Good. That's the first topic, done, out of the way. Well done, guys. Endocrine system, we flew through that. Good work. We're going to move on to the next one. So get ready. And this next one is going to be a very quick one, but it's very important that you get the terminology correct for what I'm about to teach you. Okay, give me 1 sec. Here we go. So here we go. All right, now quick one about the nervous system. Yeah, now we all know that the nervous system controls how we sense and respond to the world. Yeah, and we have something called the reflex arc, which is basically, let's quickly go to it. So we have stimulus, and make sure you know the order of this. Okay, stimulus, receptor, sensory neuron, relay neuron, motor neuron, effector. By the way, the effector, there are 2 types of effectors. It can either be a muscle, which contracts and relaxes, or it could be a gland which secretes chemicals, and then you have the response. Okay, everyone, so make sure you're familiar with the correct order of the reflex arc. Okay, now you see these neurons, sensory, relay to motor. Neurons can share information between each other, but they are not physically touching each other. A sensory neuron does not physically touch the relay neuron, and the relay neuron does not physically touch the motor neuron. So how are they able to transfer information if they're not physically touching each other? Well, that's because of a special connection called a synapse, which we're going to go through in a second. Here we go, guys. Important potential 3 marker. So if they ask you in an exam, how is information transferred from one neuron to another neuron, you're going to talk about a synapse. Okay, here we go. So first of all, let's define it. A synapse is the connection between neurons. Okay, so what we're about to go through is a synapse. Yeah, so at the end of the first neuron, as you can see on the left, or the end of the neuron, we have some chemicals that are stored at the end of the neuron, and those chemicals are called neurotransmitters. Yeah, so step 1, the electrical impulse arrives at the end of the first neuron. Now it realizes it's a dead end, there's nothing to do. So step 2, the electrical impulse causes neurotransmitters to be released from the tip of the neuron. So these neurotransmitters are released, okay? And then they travel across the gap. Now, they travel by diffusion. So make sure you mention that word there, diffusion, big mark. Neurotransmitters diffuse across the gap until they get to the next neuron. But when they get to the next neuron, you have to say this, okay? You have to say that they bind. If you don't say bind, you don't get the mark. The neurotransmitters, they bind to the next neuron, or if better, you can even say bind to receptors on the next neuron, okay? Once they bind to the receptors on the next neuron, like over here, then that makes a new impulse. There you go. So 5 points on how to explain how a synapse works. Okay, everyone? Good stuff. Wonderful. Now we're going to move on to the next topic. So that's 2 topics out of the way. It's only been 1 minute. Okay, here we go. So we're going to move on to the next topic. Guys, get ready. Stay with me. Remember, biology looks easy, but sometimes it can be a little bit hard. So here we go. All right. We're going to go through. Okay. All right. We're going to go through something called natural selection and evolution, okay? And I'm going to give you my model answer on how to answer any 6-marker natural selection questions, okay? Here we go. Let's do it, guys. So here's my 5-step formula on how to answer any question that is about natural selection. Of course, the formula is general. You have to change it based on what they ask you in the exam, but here's how you do it. Here we go. Ready? So number 1. Whenever you answer a natural selection question, you always have to start with the word variation. So in any population of the same species, like over here you can see some rabbits, in any population there's always variation. So you can see like this guy's got short ears, this one's got long ears, right? They're both rabbits. But why is there variation? Because of mutations in their DNA, okay? So you always start off your question with that. Whether it's rabbits, whether it's elephants, whether it's moths, you always start with that, okay? So as you can see here, this guy's got short ears and this guy's got long ears. Why? Because their DNA is different. Okay, number 2. Number 2, you have to talk about something called a selective pressure. So in the scenario they give you, there's going to be something that threatens the survival of that population. So it could be either maybe there's a new prey. There could be there's not enough food. Maybe there's a predator that's trying to eat them. Maybe the environment is changing and some of them can survive the new temperatures, the others can't. Or perhaps there's a pathogen that's killing some of them and not killing the rest. Okay, so you have to read the question and always pick out what the selective pressure is in that question. Okay, now the one we're going to talk about is about a predator coming to eat the rabbits. Yeah. Cool. So over here, we can say that some individuals will have advantageous alleles. So as you can see over here, there's a predator in the distance, right? So the rabbits with long ears are able to hear that, okay? So the individuals with advantageous alleles who can hear the predator, what will they do? They will be able to, what? They'll be able to survive, okay? So mention step number 3, basically, just to summarize, step number 3, you say some individuals will have advantageous alleles. You can go and expand on that if you want, you don't have to, okay? Now, the other individuals will have disadvantageous alleles and they will die. So in this case, rabbits with short ears couldn't hear the fox or the wolf, and as a result, they got wiped out, okay? And number 5, the advantageous alleles, so the rabbits with the long ears, are able to, 3 things, you have to say these 3, survive, reproduce, and pass on their genes, okay? So you always have to say those 3 things, survive, reproduce, and pass on their genes, okay? And then finally, the last point is, over several generations, okay, the advantageous allele will become more common. There you go. That's how you smash any question on natural selection. So let me repeat that one more time for those of you. So number 1, we talk about what? Variation due to mutation. Number 2, we talk about the selective pressure. So like for example here, you'd say that a wolf is going to eat the rabbits. Yeah. Number 3, we talk about some individuals will have advantageous alleles. Number 4, some individuals will have disadvantageous alleles and they will die. Okay. Number 5, the individuals with advantageous alleles will survive, reproduce, and pass on their genes, and as a result, over several generations, they will become more common. There you go. Now, any question they ask you about natural selection, whether it's about rabbits or foxes or anything, you apply this formula and you get full marks and you move on to the next question. Okay, guys? Good stuff. All right. here's a quick example. Let's just quickly do it just to apply what you just literally learned. Yeah. So natural selection can happen to even bacteria, such as MRSA. Yeah. So bacteria can also undergo natural selection. Shout out to MRSA. Okay. So here's how it works. Imagine you have a bunch of bacteria and there is variation between the bacteria because of mutation. Some of them are stronger, some of them are slightly weaker. Okay. Number two, the selective pressure is antibiotics. The antibiotic will kill the weak bacteria. Okay. Number three, the strong bacteria will survive, reproduce, and pass on their genes. Okay. Then over several generations, the stronger bacteria will become more common. There you go. Okay, guys, that's how you apply natural selection. Good stuff. Now we're going to move on to the next topic. All right, here we go, guys. Okay, now, what I'm about to go through is a little bit tricky, okay? What I'm about to go through, guys, and yes, it is in combined science. So if you're combined science, it is in your one as well, okay? So don't get scared about what I'm about to teach you. But if you don't understand, the summary at the end of the topic is all you have to memorize. Okay, guys? Here we go. Everything so far, by the way, just in case you're new, everything so far is combined science and triple, and at the end of it we'll have triple only. But this is in combined. Here we go. Genetic engineering. So let's start with a quick background, yeah? So genes, we know, are sections of DNA that control certain characteristics. For example, this eagle has a beak, it has talons, it has wings. Why? Because it has the genes for those characteristics in its DNA. This tiger, for example, over here, this tiger has paws, it has a tail. Why? Because it has the genes for those in its DNA, okay? Everything looks the way it looks because of the genes that it has. Now, in genetic engineering, the theory is this, that we can transfer genes between organisms. So obviously, this is not real, but you could potentially transfer the gene from wings from the eagle to the tiger. So what does that mean? It means the tiger potentially could have wings, right? This hasn't been done on big animals, but it has been done on bacteria. So let's talk about how it happens in bacteria. Okay, here we go. But when you do this, when you transfer the genes from one organism to another organism, okay, you create something called a GMO, a genetically modified organism. And by the way, this can also happen to plants as well. Yeah, cool. So let's talk about it. Now, the example we're going to talk about is insulin and how we make bacteria that can make insulin for us. Yeah. So look, if someone's diabetic or suffer from diabetes, they have to, like we said earlier, they have to have insulin injections. Okay. Now, in the past, guess where the insulin came from? It came from animals. So pigs and cows used to be used to extract the insulin to treat poor diabetic Dylan over here. Yeah, diabetic Dylan. Now, what's the problem with that? There's a couple of problems. Number one, side effects, right? You're using animal insulin to treat humans. Number two, religious reasons. Some religions are vegetarian or don't allow specific animals to be used to treat medical conditions, okay? Number three, ethical reasons, because you're killing animals just so that you can take their insulin, right? And number four, quantity. Look at how many millions of people in the world have diabetes, and at the same time, how many millions of animals you have to kill every year, right? It's impossible. So, because of this, we had to find a new way to make insulin. And that's where we made bacteria, okay? So we've produced bacteria, genetically modified bacteria, that can make human insulin, okay? So let's talk about this. Here's your 6 marker. Explain how bacteria can be genetically modified to produce human insulin to treat diabetes. Let's do it. Here we go. So number one, step one, and there's a summary by the way at the end of this, okay? So number one, human insulin gene is located from the DNA, yeah? And to do this, you can use any cell in the body because all your cells anyway have the same DNA, okay? So you could use a skin cell, a muscle cell, a hair cell, whatever, yeah? All right, so usually they use cheek cells because it's easier to get them. All right, cool. Anyway, you get the human insulin gene, which is just a section of that DNA, all right? Now, we have to remove it from the DNA. How do we remove it from the DNA? We have to use scissors. Not real scissors, biological scissors. And that is known as restriction enzymes, okay? So, we're going to cut out that gene using restriction enzymes. So snip, 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 snip. We're going to cut the gene out from the rest of the DNA using those enzymes, okay? Now, when that happens, notice that the gene comes out with these little Lego bits at the end of it, like little Lego or puzzle pieces called sticky ends, and that's important for later, all right? Yes, it's triplet. Yes, this is combined science, okay, guys? Moving on. All right, now we're going to grab a plasmid. A plasmid is just a ring of DNA from a bacteria, okay? So we're going to grab the bacteria's plasmid, we're going to remove it, this little nice little ring, and we're going to cut that plasmid with the same restriction enzyme that we used earlier to cut the human DNA, okay? So look, the same enzyme is going to come and only one little snip open up that plasmid, okay? Now, notice here that the Lego pieces, the sticky ends, the little puzzle pieces from the plasmid and the DNA are complementary. So, that means that the insulin gene can fit nicely into that plasmid, okay? So look, that goes nicely right there and it fits just like a puzzle. There you go, all right? Now, finally, we use the enzyme ligase enzyme to join the sticky ends, okay? So we're going to use the ligase enzyme to join the sticky ends, and that finally creates a nice little plasmid which has a bit of human DNA and specifically insulin gene human DNA, all right? So we put this plasmid back into the bacteria, and there you go. You've produced a genetically modified bacteria, okay? You've produced a genetically modified bacteria that can replicate itself and make more copies of the human insulin gene. So all you have to do is grow loads of these bacteria and then extract the insulin that's made from them, and there you go. You can use it as medicine to treat all the diabetic people in the world. Now, if that's too much for you, memorize this and that's it. Call it a day, okay? So that's literally your model answer if they ask you about how to create genetically modified bacteria. Okay, everyone? Good stuff. I'll keep it on the screen, make a screenshot, and that's it. The topic is done. Okay, I'll leave it for 10 more seconds. 10, 9, 8, 7, 6, 7, 6, 7, 6, 5, 4, 3, 2, 1. Done. Okay, moving on, everyone. Moving on to the next topic. What time is it? 9 o'clock. All right, we have time. We have time. All right, so we're going to go through vectors. That isn't vectors maths. Isn't vectors maths? Poor guy. Alright, here we go. We're going to go through genetic diagrams really quickly, okay? I know this is an easy one, but really quickly, okay? Look, if you guys think you know cystic fibrosis, in the chat, write down 3 symptoms of cystic fibrosis if you think you know it. And if you don't know 3 symptoms of cystic fibrosis, here we go. Time for you to learn it. All right, here we go. All right, so genetic diagrams, real quick, yeah? So first of all, keyword, alleles are different versions of the same gene, yeah? So you could, for example, you could have blonde hair, ginger hair, brown hair, or brunette, black hair. These are all different versions of the same gene, and the gene is hair color, okay? So these are different alleles of hair color. Now, alleles can be dominant or they could be recessive, right? Dominant is represented as a capital letter, and recessive is represented as a lowercase letter. Now, in your exam, okay, if they ask you, explain what a recessive allele means, what will you say? You can't just say lowercase letter. You could say that, yeah, you could say lowercase letter, but if it was a 2-marker and they say to you, explain what a recessive allele is, you have to say this. A recessive allele requires 2 copies, okay, in order for the characteristic to be displayed, whereas a dominant allele only requires only requires 1 copy in order for the characteristic to be displayed, okay? There you go. All right. Now, so, for example, here, if capital is brown hair and lowercase is blonde hair, so everyone's got 2 alleles. Why? Because they get 1 from each parent. Double big A is brown, okay? Big A, small a is also brown, yeah? And 2 lowercase a's is blonde, okay? Good. So we all know that now. All right. Now, genotypes can be described in 3 ways very quickly. So we have 2 capital A's is called homozygous dominant. Yeah. If you have two lowercase a's, that's called also homozygous, but recessive. And if you have a big and a small, then that's called heterozygous. All right, good, nice. Moving on, so these are called genotypes. Okay, so a genotype is what? Genotype is what combination of alleles you have, and your phenotype is what is displayed from those alleles. Okay, very quickly, let's talk about inherited diseases. Okay, so we have two examples, dominant ones and recessive ones. Let's start with cystic fibrosis. So we know that cystic fibrosis is an inherited disease. It means you get it only from your parents. You cannot be infected by it through somebody else. Okay, now, three symptoms. Number one, the lungs produce extra sticky mucus. Okay, if you have extra sticky mucus, what does that mean? It means that it will block your airways. So if it blocks your airways, you cannot breathe well. Yeah, so sticky mucus blocks your airways, but it can also block your reproductive organs and your digestive path. Yeah, so you can't breathe, you can't have children, and you can't even digest your food very well. Okay, all because of sticky mucus in the airways, in the pancreas, okay, and also in your reproductive organs. All right, good stuff. So, and obviously it's life-threatening. People with cystic fibrosis usually have a shorter life. Yeah. All right. Now, the good news though is that it's caused by a recessive allele. So, what does that mean? It means that it's less likely for you to get it. Why? Because you need 2 copies of the recessive allele in order for you to be a sufferer. Okay, so for example, if you have 2 big F's, completely normal. If you have a big F and a small f, you're still normal. You're completely healthy, but you are a carrier because maybe your potential baby could have cystic fibrosis. Okay, and if you have 2 small f's, then you are a sufferer. Okay, good. So here's an example that you could have to do in your exam. If they say to you, oh, here's parent 1 and parent 2, first of all, look, they're both normal. Yeah, they have big F, small f, they're both normal, but they're carriers. Now let's look at their children. So we make a Punnett square, which looks like this. Yeah, you put the alleles on the top and bottom like that, and then you combine. So for example, here you put big F, big F. Here you put big F, small f. Here you put big F, small f, and then you put small f, small f. Okay, and then we can see every box obviously is 25%. So we can see that what 1 out of 4 is suffering. So if 2 parents are carriers, the probability that a child will be a sufferer is 25%. Good. All right, let's move on to the next one. Polydactyly is also an inherited disorder. You get it from your parents. And what is the condition? You have extra fingers. Okay, now, is it life-threatening? No. So that's the good news. It's not life-threatening. It's not going to kill you. But the bad news is that it is dominant. So it's more likely for you to get it compared to cystic fibrosis because it's a dominant allele. There you go. That's it. Finished. Let's move on to... Hold on. There's one more thing left. All right. Okay. Yeah, that's it. Fine. Let's go. Next topic, guys. All right. So we're gonna talk about. All right, we'll come back to the other one. So we're gonna quickly go through a topic called biomass. Okay. Now this is very important because biology towards the end of it. I'll be honest, even I find it boring, but it's your last lesson, so let's just make sure we learn everything, and then after that you can burn your biology books. Okay, I'll burn it with you if you want. How about that? All right, food chains and biomass, the exciting part of biology. All right, first of all, what is a food chain? A food chain shows the direction of energy transfer in a habitat. So it shows which direction energy is traveling. Okay. So, for example, we can see energy from the plant goes to the little insects, then to the fish, and to the bigger fish. Yeah, this is a food chain. Now, we have producers, which are also known as trophic level 1. They convert light, to chemical energy. Okay, and who knows what the name of that reaction is? Photosynthesis. All right, so they convert light from the sun into chemical energy and turn that into living material, aka biomass. So if they ask you what is biomass, you just say biomass is living material. Okay, now the producers are then consumed by the next person on the chain. Okay, so biomass and energy are transferred along the chain when the organism eats. Okay, so you got trophic level 2, level 3, and level 4. All right, cool. Now this part you have to know off by heart. Okay, this number you have to know. 10% of the energy gets transferred. Yeah. So for example, if they say to you, there's 10,000 joules of energy in the first level, you have to know off by heart that the next level is going to have roughly 10% of that. So 1,000. And the next level of that is going to be 10%, so 100. And then what? 10. So can you see how every time only 10% of the energy gets transferred? So all of the rest of it, the energy, 90% of it doesn't get transferred. And they will ask you in your exam, what are 4 reasons, or what 4 reasons can you say for why energy does not get transferred? Okay, now you might know 1 or 2, but do you know all 4? Let's find out. Here we go. Okay, 4 reasons why energy transfer in a food chain is not 100%. Here we go. Number 1. So we have energy is lost as heat in the form of respiration. So when this fish, for example, eats the plant, yeah, some of the energy that it has, it uses it for respiration. Number 2, energy is used for movement. Number 3, energy is lost in waste. So even when they make poo, yeah, there's energy in that as well. All right. And number 4, not all the parts are eaten. Okay. Or you could say, for example, bones aren't eaten. There's energy in the bones, in the hair. Yeah. Not all the parts are eaten. So 4 reasons why 90% of the energy gets wasted and only 10% gets transferred. Okay. So if you guys got all of those 4, well done. You're gonna get a grade 9. If you didn't get all of those 4, don't worry. Now you know it. You're also gonna get a grade 9. Moving on. All right. Next part. The next part is talking about the producers specifically. So like the seaweed, the grass, stuff like that. Yeah. Now producers. Where did they get their energy from again? Producers, who remembers? From the sun. Okay, so the sun hits the plant and the plant gets energy from the sun. But guess what? It's not 100% efficient. So all the energy that comes from the sun doesn't actually get absorbed by the plant. Okay, and again, I know it's a boring question, but they will ask you, why are 3 reasons why the plant is not 100% efficient at converting light into chemical energy? Okay, so if you know, let's see if you get it right. Here we go. 3 reasons. Number 1, some of the energy from the sun is used to heat up the leaves instead of actually going being converted into glucose. Number 2, some of the energy is absorbed by parts of the plant that are not photosynthetic. For example, the bark of the tree or the fruit or the flower, they don't photosynthesize. Only the leaves photosynthesize. Okay, so again, energy doesn't get transferred into chemical energy. It just heats up the random stuff. Yeah. And number 3, some of the energy just goes through the leaf. Literally just goes through the leaf, comes out the other side, like the alpha particle experiment in chemistry. Yeah, it just goes through the leaf, so it doesn't get turned into energy. All right, so those are 3 reasons. Nice. Okay, now the next part, calculations. Let's quickly go over this calculation, because if it comes up tomorrow, it's going to be the easiest calculation ever. Here we go. So we're going to calculate efficiency of energy transfer. Now, some of you might know this. If you don't, let me show you. So first of all, in this new example, all right, we have the producer, which is the big tree on the left. We have the primary consumer, which is the little bugs. We have the secondary consumer, and then we have the tertiary consumer. Okay, now look, the energy of the primary consumer is 3650, and the secondary consumer is 340. Again, look, roughly 10%. Okay, roughly 10%. So the question is, what is the percentage of transfer from primary to secondary? Like, what's the exact percent? We know it's roughly 10%, but how do you calculate it? So easy. Look, all you got to do is you do the small number on top, the big number at the bottom, times 100. There. So easy, right? So easy. All right. Good stuff, everyone. Nice, nice. All right. Last bit. Okay. We have to know how to draw pyramids of biomass and pyramids of numbers. Yeah. So again, this part is elite board level knowledge. And if it comes up, I don't want you to be looking at the paper and thinking, oh man. So let's do it. Here we go. So first of all, they will ask you to draw a pyramid of biomass. Yeah. Now, what is biomass? It's the amount of material in each of these levels. And remember, keyword, very, very important. Biomass always goes down. So the first level always has the most biomass, and the last level always has the least biomass. Okay. So if they ask you to draw a pyramid of biomass, it's always going to be a pyramid shape. Yeah. It's going to be big at the bottom, It's going to be chunky at the bottom, chunky, yeah? And it's going to get smaller and smaller and smaller, like a proper pyramid, okay, everyone? Now, the way you draw it, obviously, they're going to give you graph paper, and you just have to look at these numbers, make a scale out of it, but very important, your scale has to go both directions. So if the 0 is in the middle, okay, you have to make sure that you make your scale right in the middle. So you have to go both ways, left and right, equally, yeah? All right. Now, sometimes they might ask you, yeah, so remember pyramid of biomass, yeah, like I said in the picture, always symmetrical, always at 0, and always pyramid shaped, yeah? But sometimes they will ask you a pyramid of numbers. Pyramid of numbers is different. Pyramid of numbers is about how many individuals are in each level. And we know previously, if you look at the picture here, there's only one tree, right? There's only one tree. You don't have 100 trees in this picture, yeah? So pyramid of numbers sometimes is not pyramid shape, yeah? So you have one tree which is very small, then you have big and small, etc. yeah? So the key point is pyramid of biomass always pyramid shaped, pyramid of numbers not always pyramid shaped. Good, done, easy. Let's move on. I don't want to bore you guys too much. Like I said, this is the not so fun topic, but well done, guys. You got it right. Moving on to adaptations, yeah? Real quick. Okay, listen. Triple science, we're doing that later, yeah? So don't worry about people who are saying, isn't this triple? No. If it's your first time learning this, then don't worry, okay? It's not that hard. All right, here we go, adaptations. So what is an adaptation? Adaptations help organisms survive in their environment. So without specific adaptations, organisms will not be able to survive in the ice, in the hot, in the cold, etc. Okay? So now adaptations, they fall into three categories, three main types of adaptations. You've got structural. By the way, that can also be called physical. You got behavioral, and you have functional, which sometimes is also called physiological. Yeah. So this can be called physical, yeah, in your exam, and this one could be called physiological. Yeah, but they mean the same thing. All right. So we have to look at some examples. So let's look at some. Here we go. So physical or structural adaptations are features of the organism's body that helps it to survive, part of the body, okay? And I'll give some examples. Next, behavioral is how it behaves, an action or a way an organism behaves that helps it to survive. So it does something which makes it survive. And then finally, you've got functional, which is internal processes or functions that helps the organism to survive, yeah? So something that's happening inside its body, a reaction or a specific process that's allowing it to survive. Okay, let's do some quick examples. Here we go. Let's start with the elefante. Yeah. So elephant has a long trunk. We know that long trunk is part of its body, so it's a structural adaptation. And what does it do? It helps it to grab food and water, and sometimes tourists if they're too close to them. Yeah. Large ears, again, part of the body. Large ears. What does it do? It increases the surface area so that it can lose heat and cool down because it lives in a hot place. Yeah. All right. So, and finally, wide feet. Why? Because the wider the feet, the more it can distribute its weight, so it can walk across long distances, from stand to barking, for example. All right, next part, behavioral. So an example of behavioral is that it sprays itself with water or mud to help itself cool down. Yeah, so it's acting in a certain way. Or, for example, it can go and sit under the shade, and that helps it to cool down. Okay, everyone. So I hope you know that that's behavioral. And finally. What are you guys writing in the comments? Finally, functional. Here's an example of functional. So, elephants can control the size of the blood vessel. Obviously, not consciously, but unconsciously. The blood vessels in their ears can get bigger and smaller, increasing the blood flow to their ears. Why would it do that? Well, if it's feeling super hot, it will increase the blood vessels, size of the blood vessels, so that way more blood can go to the ears, and then that way more heat can escape the elephant. So that's an example of functional. And another example of functional is that not just elephants, even camels, their kidneys produce concentrated urine. Okay, and what does that mean? It means that it reabsorbs all of the water, so that way it doesn't become thirsty or dehydrated. So the urine comes out super concentrated. Why? Because all of the water remains in the body of the elephant. All right, there you go. Now there's more examples, but I think you guys get the gist of it, so I'm not going to go through any more examples. But yeah, that's adaptations. Good work. Done. Smashed another chapter. Okay. So let's do a quick practical, yeah, because I know you guys love practicals. Especially, I know you guys love the one about the little square frame. Shout out to Quadrats. All right, here we go. So, what is a quadrat, first of all? A quadrat is a frame that's 1 meter by 1 meter. Yeah, the area of that is 1 meter squared. Now, why am I being so specific about this? Because once I saw in a past paper, they tricked the students by saying Susie had a different quadrat, which was 0.5 meter by 0.5 meter. Okay, so the area of that quadrat wasn't 1 meter squared, it was 0.25 meters squared. Okay, so you have to just make sure if they give you a tough calculation, you always have to times the numbers together to get the actual area of your quadrat. Yeah, but anyway, that's just a little warning from me. All right, but anyway, what's the point of a quadrat? What do we do with it? We use a quadrat to count the number of individuals in an area. Yeah, for example, if I said to you, go count the number of flowers in the park because you want to pick them up and give them to your biology teacher, right? It's very long to go and count every single flower in the park. You won't have time, okay? But instead, what we can do is we can estimate the number of individuals by doing the following. Here we go. Step 1. So what's the question going to be? In the exam, the question will be, it will be, describe an experiment where you can estimate the number of roses in the park. Something like that. Yeah? So here we go. So step 1. Step 1 is draw a map of the park. That's easy, step 1. Step 2 is draw a grid on the map. Okay? Make a nice little grid on the map. 2 marks already, by the way. Just letting you know. Yeah, 2 marks already. Step 3 is select random points on the map using a random number generator. Okay, so you don't have to do this in your exam, but on your calculator you can actually get random numbers by pressing the random button. Yeah. All right. Step 4 is now you actually go in real life to the park and you go to those places that you selected, those coordinates, and you place your quadrat on those coordinates. Okay. Step 5 is count the number of daffodils in each quadrat. So there's 6 in this one, there's 4 in that one, 7 here, 3 here, 5 there. But quick question, by the way, if one of the quadrats had 0 daffodils, would you still count that as one of your readings, or would you ignore that? You would count it, yeah? All right, good. All right, and then finally, so once you got like 6, 4, 7, 3, 5, then what you have to do is remember these are all per meter squared, right? These are all per meter squared. 3 per meter squared, 5 per meter squared, 4 per meter squared, yeah? 6 per meter squared. But we want to know the average. So how do you get average again? You add them up, right? You add them all up and you divide by. In this case, you're going to divide by 5 because there are 5 quadrats, yeah? So you add up, divide by 5, and you get your average per meter squared. So here we got, obviously that's a wrong number, it shouldn't be 2.5, but yeah, so let's pretend it was 2.5 daffodils per meter squared, okay? Then final part is, take this number, so here's your average that you got, and multiply it by the area of the entire park. So if they told you that the area of the park was, let's say, 600 meters squared, and you got 2.5 as your average, you times it together, you get what? You'll get 1500, and that's how many daffodils you estimate are in that park. Okay, everyone? So look, here's a summary on how to use quadrats to estimate the number of individuals in a certain habitat. All right, so screenshot that if you want, and we can move on to the next practical. All right, here we go, guys. Is everyone ready? Okay. Sorry, one sec. Yeah. All right. Next part, everyone. Ready? All right. So now we're gonna do something called yeah, that's fine. Thanks. All right. Next part. We're gonna talk about what? Transects. Okay. So transect is a bit different because transect is not about estimating the number of individuals. Transect is about estimating the what? The distribution. So for example, are there more daffodils on this side of the park or on this side of the park? And if so, what factor is causing there to be more daffodils here compared to there? Yeah? All right, so what we're going to do is quickly go through that, and then we'll have a short break, everyone. We'll have a very short break after this, and then we'll continue, okay? So again, let me say the question. The question is, Describe an experiment where you can investigate the change in distribution of daffodils across Stanmore Park. Yeah? Here we go. Here's what you do. Number one is make a transect using a tape and 2 poles. What is a transect, by the way? What you see on the screen, those 2 poles and that tape, that is called a transect. Yeah? That's called a transect. So number 1, make a transect across the park. Number 2, place quadrats along your transect. So can you see over here, there's a quadrat. You're going to place your quadrat from there, and you're going to start by counting. So you're going to count the number of flowers that you're investigating in that specific position, and then you flip your quadrat. You flip it along, count, flip, count, flip, count, flip, count, flip, count, flip, count, flip, count. Flip, count, flip, count, and you do along the entire transect. Okay, so you can see here that the number of daffodils are not what? The number of daffodils are not the same. You can see there's more on the right compared to on the left, and in the middle there's hardly any. Okay, now at the same time you want to investigate another factor. So the question could say the pH. So what would you do is, while you're counting and flipping, you also stick in your pH probe into the ground, so you can record an abiotic factor. What does abiotic mean again? Abiotic means non-living factor. So you stick your pH probe into the ground and you measure the pH while you're also measuring the number of individuals. Okay, and that way you can compare. You can see, oh look, when the pH was higher, there's more daffodils. So that shows there's a correlation between pH and number of daffodils. There you go. So easy. Okay, good. Is there a summary? Oh, sorry, one more thing. It's not just pH. Other factors could be light. So you can use a light meter, or moisture. You can use a moisture meter. There you go. So there's your summary on how to. There's a summary on how to explain how to measure the distribution of organisms using a transect. Okay, be right back in 2 minutes. This is going to chat to you for a bit.
We have a break right now because it's very intense. And what you need to do in your break is, if you need to pray Maghrib, then just pray now. But other than that, other than the break, if you guys are still watching, exam season is coming to an end. You've only got 2 weeks left, and in these 2 weeks you really need to lock in. Okay, I mean, if you haven't locked in by now, that is quite concerning, but at the same time, there is so much going on for you guys, which we understand. There's so many exams happening, big ones, small ones. You got your maths, you got your English, you got your sciences, and with 2 weeks left, I think a lot of people, what happens is the first few exams go quite bad, and they think that, what's the point of even trying in these paper 2s, because my paper 1's gone bad. You actually should have the opposite mindset. If you did bad in paper 1, this is how you make up for your grade, by smashing paper 2. Okay, you smash paper 2, and believe it or not, that will make you do better than you originally thought you would have got. So maybe you were aiming for a 6, right, or a 7, and you flop paper 1, you're thinking, I'm just gonna get maybe a grade 3 or 4, like as long as I get a grade 5, I'm fine. Rather, if you smash this paper 2, right, you watch this live stream, you smash this paper 2, you can actually get better than what you were originally planning, which was the grade 6 and 7. You can actually get grade 8 or 9. Okay, guys, to help with this, what we've done is the moderator, the admin, has put a code in the chat. Use code livestream40 and our exam season package, okay, which is normally, look at that, it's normally £130. Actually, it's actually normally £149. But this exam season package, we made it £59. But if you use this code right now, livestream40, okay, because we're in a live stream right now, so only the guys in the live stream can see this code. Use livestream40, add promotion code, and what happens is, so watch this, livestream40. Okay, you can do it after the live stream's finished, or you can do it in this break now, okay, whilst we have the break. This goes down to £19. For £19, you see the slides that Mohammed was just using, right? You get access to all those slides for biology paper 2, for physics paper 2, and for chemistry paper 2. Okay? All those resources that you were just looking at, okay, you get them for yourself. So rather than using them and relying on the night before live stream, okay, you've got a whole week. You guys told me, right? You got physics paper 2 on not this Monday come out, the next Monday come out. If you buy the slides now, okay, after this live stream or during this break, you can use, so look at this. You can use this platform where we have all the slides that Mohammed was using, but the physics equivalent and the chemistry equivalent for the entire week, okay? And we've got AQA, Edexcel, OCR. It's literally on a plate for you guys, okay? So can you see all these slides? And we've got corresponding videos as well. Use it for this week and smash your paper 2s, okay, for sciences, and of course your paper 3 for maths. Does that make sense, guys? So look, of course, paper 1 content is already done, so we've already sectioned that nicely for you guys as well, so that you don't need to look at paper 1 content. You can just scroll, doom scroll, all the way to, bang, paper 2 content. So you can skip all the paper 1 stuff, go straight to paper 2 content, and just bang out the slides for all of those, bang out the worksheets, bang out the videos, okay? And really consolidate your remaining exams, guys. So that's all from me, Coach Livestream 40. Our exam season package goes all the way down to £19. I mean, that is a bargain. If that's not a bargain, I don't know what that is.
Bang. Wow, thank you very much, Ishan.
No
Is it only working out until this live stream?
The thing is that we might discontinue this code after the live stream is finished, but maybe 2 hour window, because, you know, they need to get their card and stuff. But after midnight,
yeah,
it might be done.
Maybe I'll get myself.
After midnight, it might go back up to £59. So 19. If you wanted £19, use it now or after live stream is ended, guys.
Fairs. Well, generous Ishan. W in the chat for generous Ishan. Okay, so everyone, we are back and we shall continue with biology. Thank you for coming back, and stay tuned. We are about to smash the next topic. Absolutely. Okay. Hold on, everyone. Hold on, everybody. Okay, so we're gonna do just 2 more topics, and then after that, combined science. You guys may leave after that, okay? But until then, just stay with me because I need to teach you quickly a couple of important stuff. Okay. So this part is really important, yeah, which is not super important, but just a very short side topic, yeah. So let's say you have an animal. So here's a specific animal, yeah. This animal in different languages has different names. So English is called lion, in Farsi is called sheer, Bangla is called bhag, Arabic is called asad, Spanish is called leon, German is called Louis, yeah. Now, imagine if all the different scientists in the world used their home language to speak about the same animal. What would happen in scientific literature? What would happen? We would all get confused, right? If an Arabic scientist is reading a German paper written by a German scientist, and like, oh, what's a Lewy, right? They're going to get confused, okay? So in scientific literature, we don't use our home or our native description. We use a different way of naming things, and that is called the binomial system, okay? So the binomial system is a universal way of naming all living things, including human beings as well, yeah? Now, where does it come from? It comes from two things. It comes from the genus and then the species of an organism. So for example, and why do we do this? Mainly to avoid confusion between scientists, okay? So a lion, for example, is not called lion in scientific literature. It's called Panthera leo, yeah? So the Panthera is the genus and Leo is the species, okay? And that can be shortened to P. leo. So here's another example. So this one is called Panthera tigris, right? Panthera is the genus and tigris is the species, and it can be shortened down to P. tigris. So a question they could say to you is, and for humans, by the way, what is humans? Homo. So Homo is our genus, Homo, and then sapiens is our species, and it's H. sapiens as the shortened version of it, okay? So here's four animals. Which ones would you say out of these 4 animals are the most closely related? You can tell just by looking at the name, okay? So if you look at the names, we can see that A and C, right? They have the same genus, whereas B and D have different genuses, okay? So A and C have the same genus, but they are different species. So we can say that A and C are more closely related to each other than the other animals, okay? And by the way, these are horse, cat, zebra, and dog, okay? There you go. So that's called the binomial system. It's how you name organisms in scientific literature, and is used to prevent confusion and help to identify who they are, and also to see who's closely related. All right, moving on to the next one. Okay, guys. So we have one or two more topics left, and after that, I'll leave it to you. Okay, this one's super duper important, okay? Meiosis. Now, I know you guys know meiosis already, but I'm just going to show you animation-wise what it is, and then after that, I'm going to give you a very important table that I want you to memorize, okay? So for now, just watch my cool animation of how meiosis works, okay? So here we go. Meiosis, step one, and you don't need to write this down, okay? Because it's going to be long for you to write it down. Number one, the meiosis. Chromosomes line up in the middle in pairs. Step one, okay? They line up in pairs in the middle. Then, step two, the centrioles pull the chromosomes apart. So the pairs get pulled apart in step two, yeah? Then nucleus forms around the chromosomes and the cell divides. So that's the first division, okay? So what was it? Chromosomes line up in pairs, centrioles using spindle fiber pull the pairs apart, and then cell divides. But then the chromosomes, one more time, they line up in the middle, but this time not in pairs, okay? They just line up in the middle. And once again, the centriole pulls apart the arms, not the pairs, but this time it pulls apart the arms. And the arms are known as sister chromatids, yeah? All right, so the sister chromatids get pulled apart, and then once again, the nucleus forms and the cell divides. And you get 4 cells at the end of it, okay? Now, each of these cells are referred to as haploid cells. What does that mean? It means if a normal body cell has 23 pairs of chromosomes, right? And we've called those diploid, we call that a diploid cell, then the haploid has only 23 chromosomes. Okay, guys? And that's for humans, but for different organisms, it could be different. So for example, let's say an onion has normally 56 pairs, for example. That's not true. The diploid is 56 pairs. The haploid will be only 56. Yeah? All right. So this table is super important, okay? Comparing mitosis and meiosis. So in meiosis, as we saw, there are 2 divisions, whereas mitosis only has 1 division and it's finished. Meiosis has 4 daughter cells, whereas mitosis only makes 2 daughter cells. Okay, in meiosis, all of the daughter cells are haploids, whereas in mitosis they're diploids. Yeah, they have the full set in mitosis. Okay, mitosis is used to produce body cells, so skin, hair, muscle, you name it, except for gametes, sperm and egg are produced by meiosis, okay? And one more thing which I haven't written, in mitosis, all the cells produced are identical. So the daughter cells are identical, whereas in meiosis, the daughter cells there is variation between the daughter cells because of something called independent shuffling or random shuffling, okay? Good. So that's the comparison between meiosis and mitosis. I hope you guys got all of those points. Now let's move on to our final few topics before we say good night to combined science and end your final biology lesson ever. Okay, so let's have a look. We done natural selection. Let's see. One sec, everyone. Oh, how can I forget such an important topic? Menstrual cycle and what? Fertility. We have to go through this, guys. We have to do this. All right, here we go. So, let's quickly talk about the menstrual cycle, yeah? So, we know that men have testosterone, which is produced by the testes, and that controls male characteristics, whereas females have estrogen produced by the ovaries, and that controls female characteristics, okay? But on top of that, females also have something called a menstrual cycle, which is basically a cycle that repeats every 28 days in a process called ovulation, where an egg gets released from one of the ovaries, okay? But we have to know which hormones which hormones and the order of them, okay? So here we go. So the 4 hormones are estrogen, FSH, which stands for follicle stimulating hormone. You can just call it FSH. LH, luteinizing hormone, and progesterone. Okay, so we have to know these hormones and what they do. Now, from these hormones, who knows which one is the first hormone that comes in the cycle? Here's a clue. F for FSH, F for first. So FSH comes first, okay? Then estrogen, that comes second, and then LH comes third, or you could say last out of the three, and progesterone is the little final cherry on top, okay? So first is FSH, then it's estrogen, and then LH comes last, and then progesterone. All right, now let's talk about this in detail. Here we go. So step 1 in the menstrual cycle. FSH is produced by the pituitary gland, yeah? So it's produced up here in the pituitary gland, and it doesn't stay there. It travels down all the way to the ovaries. So it goes from the pituitary gland down, down, down until it gets to the ovaries, okay? Now, what does it do in the ovaries? It does two things, okay? Number 1, it makes a follicle, which is a baby egg. It makes it mature into a mature egg, yeah? That's one thing that FSH does, like go, grow, grow, grow. And number two, it tells the ovaries to produce the hormone estrogen, okay? So it comes from the pituitary gland, goes to the ovaries, it says to the follicle, grow, and it says to the ovaries, produce estrogen and go. And estrogen goes back up all the way back to the pituitary gland, okay? So estrogen, by the way, one more thing. So now what does estrogen do? Two things. Number 1, it causes the lining of the uterus to build up, yeah? So the uterus over here, the lining gets a bit thicker, okay? Why is it getting thicker? Randomly? No, not randomly. Because it's expecting a fertilized egg to be landing there very soon, maybe, yeah? That's why it's preparing itself. And then number 2, estrogen travels up all the way to the pituitary gland, where it says, stop making FSH, and now make LH, okay? So what does estrogen do? It goes up to the pituitary gland and says, stop FSH, send down LH, okay? So LH comes down from the pituitary gland, back to the ovaries, okay? And by now, remember that little baby egg, the follicle? By now, it should be ready, it should be mature. So LH, when it gets down to the ovaries, the follicle, which has matured over time, thanks to FSH, is now ready. So all it's going to do, it says, go, release. It's going to release it into the fallopian tube. So it's going to travel along the fallopian tube. Now, while this happens, while it's traveling along the fallopian tube, maybe it might get fertilized, maybe not. We don't know, yeah? Depends on what happens. But let's see. Progesterone is also involved, and that maintains the lining of the uterus for 2 weeks, okay? So progesterone maintains the lining of the uterus for 2 weeks there. All right. And then also it stops more FSH and LH from being what? Being released. Okay. Now, finally, if the egg is fertilized on its way to the down the fallopian tube, the uterus lining stays there for 9 months until the baby is born. But if it is not fertilized, if the egg does not get fertilized, then the uterus lining and the egg both come out of the body, okay? And the whole breaks down and comes out of the body, and then the whole cycle restarts in 2 weeks. There you go. And here's a nice little summary of the entire menstrual cycle. If you guys like to read summaries, there you go. Perfect. Done. Okie doke. So I am going to now you guys are actually so crazy, yeah? Some of you comment, I don't know what you guys are writing in the chat, but that is mad. All right, let's quickly, somebody requested the carbon cycle, and I'm not going to say no to requests because I want you to do well. And since it is your last biology lesson, I shall comply. So let's do the carbon cycle. Give me a sec, yeah? Looking for it for you right now. Okay, here we go. Carbon cycle as requested by one of you. All right, here we go. Carbon cycle, important one. Here we go, everyone. Ready? Here we go. All right, so number 1, the carbon cycle starts with carbon dioxide in the air, okay? So carbon dioxide from the air gets turned into carbon compounds such as glucose by photosynthesis. That's step 1, yeah? CO2 from the air gets absorbed by the plant and gets turned into glucose by photosynthesis. Okay, now the plant also respires. So some of the CO2 gets released back into the air by respiration. So coming in by photosynthesis, going out by respiration, yeah? Now, what else happens to the plant? Well, some of the carbon goes to animals. Why? Because when the animal eats the plant, the carbon from the glucose goes into the animal by feeding, okay? So now some of the carbon that used to be in the air went to the fruit, and from the fruit, it went to the animal, okay? What do animals do? Respire, right? So animals, when they respire, they also release carbon dioxide. Okay, so in by photosynthesis, from the plant to the animal by feeding, and plant and animal both respire, releasing carbon dioxide into the air. All right. Now, when the animal dies, or when the plant dies, both of them, carbon dioxide is released by decomposition. So little microbes, they eat the animal and plant, and as they do that, they release carbon dioxide. But is the entire animal eaten by the microbes? No, because the bones remain. So the bones go down into the ground over millions of years and they turn into fossil fuels, okay? So remember, all the carbon that was once in the air, then into the fruit, then into the animal, went into the bones, and from the bones it went into the fossil fuel. And what happens when we burn fossil fuels? Again, we release carbon dioxide by combustion, okay? So that is the carbon cycle in a nutshell. All right. Sorry? No, still not finished. Okay, carbon cycle. Nice, done. Okay, next topic. We've done this, we've done that, we've done this. This, done. Oh, it looks like we finished pretty much most of the stuff in combined science. No way. That's done. Okay, guys. Alright, combined science. Okay, combined science. We have concluded your biology lesson. Well done. From year 7 to year 11, you guys did biology for 5 years. Every single day, biology. And tomorrow's your last biology exam. And after that, you're free from biology. So thank you very much for tuning in. If you're combined science, you may now go to sleep.
They don't need to buy anything, but they can give us a sub, so we'll get 100k.
Oh, but guys, come on. What are we on?
91. So
We're 91k subscribers on YouTube. Come on, guys, smash that sub button.
just support us by subscribing.
Yeah, support us so we can get a YouTube plaque. Oh, it's so sick. All right. Anyway, I'm going to move on to triple science. So, what's it called? Let's go through that. Yeah. Those of you who are saying peat bogs, it's not coming up. It's not coming up. Trust me. Don't worry about peat bogs. It's not coming up, mate. All right. Listen, triple science. We got a lot to go through. So, goodbye, combined science. Have a good sleep. Yeah, we're gonna come. We'll be finished soon. All right. Here we go. Correcting vision defects. Well, you guys got bad, man. All right, here we go. Correcting vision defects, triple science. Okay, so first of all, we know that the lens in the human eye is a convex lens, yeah? It's a convex lens. So this lens, every single human being has a convex lens. What does that mean? It means when the light hits the lens, it comes in, and it hits the back of the eye, which is known as the what? The what? The back of the eye, yeah? All right. Now, it's called the retina. I had a little brain freeze moment there, yeah? It hits the retina, and if it hits the retina properly, exactly like that, you can see good. But sometimes the lens in your eye is not working properly, so either the light, that's called the focal point, by the way, yeah? So either the light will come closer or further away, and because of that, you get blurry vision, okay? Now, some people, for example, some people have something called hyperopia. What does hyperopia mean? They are long-sighted. What does that mean? It means they can see far away very well, but they cannot see things that are close to them. They are long-sighted, okay? Now, why are they long-sighted? Because of this. When someone is long-sighted, either they have a short eyeball or their lens is too weak. It's too weak at bringing the light. It takes forever to bring it in, okay? So in either case, the focal point is behind the retina. Not good, yeah? We want it to be on the retina. But this little lens over here is not good, is not strong enough to converge the light properly, okay? So, How do we fix that? You go to the optician. They're going to give you another lens. They're going to give you another what? Convex lens. Okay. So for hyperopia, we give the person a convex lens, and what that will do is, if you look at this picture, look at the red line and now look at the blue line. See? So when the light hits the first lens, it comes in a little bit. When it hits your eye, it comes in much more, and then eventually it'll hit the retina properly. Okay? So the convex lens brings the focal point closer, and it hits, so that way it hits your retina properly. Good. All right, moving on. If someone is short-sighted, they have myopia. So what does that mean? It means they can see close very well, but they cannot see far very well. So for this person, their lens is too strong. Or they have a very long eyeball. Yeah. So either the lens is too strong, their convex lens in their eye, or their eyeball has a long shape, like kind of like a rugby ball. Yeah. So what does that mean? It means that the focal point is too close. It's way before the retina. Yeah. How do we treat this person? We give them a what? We give them a concave lens. So what the concave lens will do is it will push away the light, and then it will hit the lens, and then it will move it backwards, so that way it can hit the retina properly. There you go. Okay. So that's how we treat what? Eye conditions. All right. Good. Moving on. Next topic. Next topic, guys. We ain't got time. Alright, kidney. Let's do kidney real quick. Hey, you guys want to do kidney? Put K in the chat if you want to do kidney. Alright, fine, we'll do kidney. Alright, here we go. So, here's your kidney, okay? Now, in your kidney, blood containing waste enters the kidney from a special blood vessel called the renal artery. So the renal artery brings blood into the kidney, okay? Now, this is not clean blood. This is blood that contains waste, yeah? Particularly, what's the main type of waste? Urea, yeah? Urea and ions, excess ions, and obviously excess water, yeah? Anyway, that comes to the kidneys from the renal artery. The job of your kidneys is to filter. So what it's going to do is the waste goes into the ureter, which goes to your bladder. Okay, it goes to your bladder and then forms urine. And the clean blood, the one without the waste, goes to the renal vein and then goes back to your body. Okay, guys. So waste containing blood comes in by the renal artery. The kidney filters it. The waste goes out the ureter to your bladder. Okay, and the clean blood goes out back up using the renal vein. Yeah. Now let's quickly talk about how this happens inside the kidney. Like, what's going on? How is it even doing this? So let's zoom in. Yeah. So number one, here's what happens in the kidney. So you have this red part again. This is part of your renal artery. Okay. Remember we spoke about that earlier. And then this green thing, this is called a nephron. It's part of your kidney. It's part of the filtration system in your kidney. It's called a nephron. Okay, so here's what happens when blood enters the kidney from the renal artery. When blood enters the kidney, okay, it gets squeezed. The blood gets squeezed, and this process is called ultrafiltration. It gets squeezed. Yeah. Now, when it gets squeezed, small components of the blood get squeezed out of the blood and they go into the nephron. So those are glucose, yeah, ions, water, and urea. They get squeezed out of the blood into the nephron. Large molecules like cells and proteins do not leave the blood. They stay in the blood. They're too large. Okay, so that's step one. What was step one? Ultrafiltration of the small components out of the blood into the nephron. Okay, now glucose and water, like, that's good, right? We don't want to get rid of that from our body, do we? We want to keep that. Okay, so what happens is in step two, as the fluid is traveling on its way to the bladder, okay, what happens is some of the molecules that got squeezed out earlier by accident, just because they were very small, they get reabsorbed. So glucose, for example, and some of the water, not all of it, some of the water, they get reabsorbed, and that is called selective reabsorption. Okay, they get reabsorbed, and urea and the other excess water, that does not get reabsorbed, and that goes out and that forms your urine. Okay, so that's how it works. All right, now the volume and concentration of urine can differ. Like, for example, have you ever had a day where you drank loads of water, you went to the toilet, and you produced lots of urine and very dilute urine, right? And if you didn't drink water much that day, the amount of urine that you had was low and very dark, okay? Why is that happening? It's because of hormones. So if you have less water in your body, your kidneys make less urine. If you have more water that day, your kidneys make more urine. And all of that is controlled by negative feedback and a hormone called ADH, okay? So let's talk about it. So what happens when there is not enough water in the body? Yeah? So what's the question? The question is, describe how hormones in the body control how much urine is produced. Yeah? That's the question. Here's the answer. Number one, Number one, receptors in the brain detect the low water concentration in your blood. So your brain already knows that you're dehydrated. Yeah, it already knows about it. What does it do about it? So step two, it coordinates a response in which it instructs the pituitary gland to make the hormone ADH. Okay, not ADHD. ADH, anti-diuretic hormone. Yeah, so the brain is like, oh, I'm thirsty or I'm dehydrated. Okay, pituitary gland, make ADH. Go on, make some ADH. Yeah, so the pituitary gland, when it makes ADH, right, it sends it in the blood. Remember, it's a hormone, right? It's a chemical messenger that travels in the blood. It sends it in the blood all the way towards the what? Towards the kidneys. Okay, so as it travels towards the kidneys, what do you think it's going to do? It's going to tell the kidneys to what? Is it going to tell the kidneys to release more water out of the body, or is it going to tell the kidneys to keep in more water because we're thirsty? Which one? It's going to say what? Reabsorb more water. Don't turn it into urine. Reabsorb it and send it back to the body because the body is already low in water. Yeah, and as a result, what happens to the amount of urine in the body? Well, since all the water is going back up, very little water is going to come out. So your urine becomes what? Your urine becomes low and concentrated. Okay, here's a summary, guys. Here we go. So low water, more ADH, more water reabsorbed, less urine. And the opposite, if you have lots of water, less ADH, less water reabsorbed, because you already have so much water already in your body, so it's not getting reabsorbed, it's just coming out the body. Lots of dilute urine. Okay, good stuff, everyone. All right, nice. Okay, very quick one, yeah? What happens if the kidneys stop working? So if the kidneys stop working, you have something called dialysis, and you also have a kidney transplant. But let's quickly look at dialysis very, very quickly, yeah? So in dialysis, here's what happens. Number one, remember, this person's kidneys doesn't work. It cannot clean the blood. So the person has to go to the hospital, or they have a machine at home. The blood from their body goes into the machine, the blood that contains waste. The machine cleans it, it filters the blood. It does the job of the kidney by removing the urea, okay? Removing the excess ions. And then the clean blood returns back to the person's body, yeah? So that's what dialysis is. It cleans the blood for the person. So how does it work though? This part is quite important. It's a little bit detailed, but let me tell you, yeah? So first of all, to put it simply, okay? Number one, the patient's blood goes into the machine. So look, here's the blood coming from the person, yeah? Is this clean blood or not? If it's coming from out the person. Is it clean? Nope, it's not clean, you're right? Because it has waste, yeah? Also, the machine has a partially permeable membrane. Because of that, we can have exchange of substances, like the waste can leave the blood and go to the fluid around it. If there was no membrane, you cannot have exchange, yeah? All right, so around the blood, we have something called dialysis fluid, which is clean, healthy fluid, okay? It has no waste in it. All right, so notice that the amount of glucose in the fluid and the blood is matched. Why? Why do you want to have the same amount of glucose in the blood and the fluid? Because you don't want any glucose leaving or entering, yeah? You just want it to stay constant, because remember, it's not about glucose, it's about urea. So, number two, there is no, can you see how there's so much urea in the blood, right? From the person, all this urea, all of these ions, all of these water, okay? So, because there's so much of that inside and there's nothing in the fluid, it's going to leave by diffusion. So, all the urea and the excess ions are going to leave the person's blood and they're going to go into the fluid, contaminating it, okay? But look what happened to the blood. It got clean. the blood got cleaned up because all the urea left, okay? So the waste diffuses out, the blood gets cleaned, all the urea has left, and the blood goes back to the person, okay? It goes back to the person. But we have to make sure that before the next batch of blood comes, that the dialysis fluid itself also gets refreshed. If we don't clean the fluid, then if the blood comes again, what could happen? The urea could go back inside the person's body, isn't it? Yeah. So we have to make sure that the dialysis fluid is refreshed regularly to prevent any urea going back into the person's blood. There you go. So summary of dialysis. There you go. You can screenshot that. If they ask you how does dialysis work, you have the best answer right there in front of you. All right, done. Screenshot that. We're going to move on. Okie doke. So that's done. That's done. Moving on to this one. What do we have here? Moving on to the next one. All right. There's a bunch of topics, but let's just quickly go through. All right, we're going to go through cloning, guys. Okay, cloning. That's very important. Oh. Yep, everything's fine. What about you guys? Nice. Okay, so we're going to talk about how to clone animals. Yeah, animal cloning. So first of all, there's two types of cloning. There's two ways, that you can clone an animal. One is called adult cell cloning, and the other one is called embryo splitting. Okay, so let's start with adult cell cloning. So let's say you have your favorite animal in the world. Let's say like a favorite horse. Okay, this horse is so beautiful, it has the best hair, and you just want another horse, okay, of the exact copy of it. That's when you do animal cloning. Yeah, so you take a adult cell. So you take your horse, you take it to the side so no one's watching, and you just grab a cell out of its body. Yeah, now from that animal body cell, you remove the nucleus. Okay, now remember, because it's a body cell, the nucleus is going to be a diploid, right? It's going to have 23 pairs of chromosomes. Well, it's going to not 23 because it's a horse, but whatever, but pairs. It's a diploid. It's a proper nucleus. Okay, then you're going to get a egg from a female horse. So any female horse, even like the ones you don't like, you grab it, you get the egg, okay, but you remove the nucleus from the egg. We don't want its nucleus. We just want the egg itself. Yeah. So you throw away the nucleus. So what you have now is an egg without a nucleus, okay? But it's okay because we already have the nucleus from our favorite horse ready. So we put that nucleus into the egg. Now what we have is basically a zygote, okay? So this is called a zygote. Why? Because it has a full set of chromosomes, right? Pairs. Then we zap it with electricity, okay? We give it a little electric shock, and that will start the process of mitosis. So it will do mitosis after the zap. So it will divide every time by 2, by 2, by 2. By the way, there was a question once, yeah, in the exam. It says you start with a zygote. Yeah, it says you start with a zygote, and it does mitosis 3 times. How many eggs do you have after 3 cycles of mitosis? Well, let's think about it. If you start with 1 and it doubles, that becomes 2. And that doubles, that becomes 4. And that doubles becomes 8. Okay, so that was a question. Just bear in mind it's mitosis times 2, not times 4. Yeah. All right. Anyway, so now what we have here is a bunch of cells, identical cells. Yeah. This is what it's a small embryo. Okay. What do we do with this embryo? We take it, and once it becomes an embryo, which is around 16 to 32 cells, you grab this little embryo, and then you're going to what? Place it inside a surrogate. So you're going to take a female horse and place the embryo inside the female horse, and then it will give birth. The baby that's born will be a copy of your favorite horse. It will not look anything like the mom. It will not look anything like the surrogate or the mom that you got the egg from. It will be an exact clone of the horse that you got the nucleus from. Okay, that's called adult cell cloning. All right, moving on. Let's talk about the next type of cloning. This one is called embryonic splitting. Okay, so let's say we have a very strong male cow and a very strong female cow. Okay, and I want from them 20 baby cows that are all the same, all identical, 20 baby cows. they're identical to each other, but not to the parents, okay? How do I force them to have all these baby cows identical? By doing something called embryonic splitting. So, step one, you're gonna take the sperm from your strong male cow and the egg from your strong female cow, okay? And then you're not going to remove any nucleus in this one. No, don't remove any nucleus. All you're going to do is you're going to fuse them together, okay? When you fuse them together, you're going to get what? A zygote, okay? So remember, if this is this cow, this is Bob, okay? And this is Betty, yeah? The female cow Betty. This zygote, is it a clone of Bob? Is it a clone of Betty, or is it different? It's different. So this one's called, for example, like Billy, yeah. So it's gonna be neither like its mom or like its dad. It's completely different, yeah. All right. Then you allow that cell to multiply until you have an embryo. Okay. Then we grab the embryo and we split it into smaller bits. So kind of like when you're making pizza, you have a big dough ball. You move it into smaller dough balls, yeah. Okay. And then, yeah, okay, fine. We will do practical decay, everyone. No problem, yeah? Lock in. So, okay, lock in, yeah? All right. Then you, what's it called? Yeah, so then you have these small embryo balls. But now, here's the thing, right? These embryo clusters that you took from the original one, these are the clones, okay? So the clones here are the children. They're exact clones of each other. And then you put them into the surrogates, and the babies that are born are going to be clones of each other. So they're going to look like each other, not like their parents, a mixture of both parents, but the cloning here is that the children will be like twins, yeah? Or you can have more than twins. If you want to have like 10 children, you can split the embryo into 10 little pieces and put it into 10 different females and have 10 identical clones. There you go. That's how you do cloning. Good work, everyone. All right. We will do the practical decay, practical thing. Okay, let's go. Let's do practical since you guys like that one. All right, we're gonna do investigating decay practical. Okay, everyone, lock in for this one. Yes, you. Someone says, "Can I clone myself?" You can clone yourself if you want. If that's what you're about, why not? Yeah, we can do fossils too, no problem, and we can do protein synthesis. Yeah. Okay, lock in time. All right, so investigating decay. All right, so. Here we go, everyone. Lipase breaks down fats into fatty acid and glycerol. So imagine if you have this is a lipid, okay? A lipid gets broken down by lipase into what? Fatty acid and glycerol, okay? Side point. As this happens and you're making more fatty acids, what will happen to the pH of the solution of the reaction? Well, we know acids have a low pH, right? So as you make more and more fatty acids, the pH of the reaction is going to go lower and lower and lower, okay? Bear that in mind, just because you're releasing all these acids from the lipid, okay? Anyway, moving on. So what we can do is we can investigate the effect of temperature on the speed of this reaction. We can investigate that, and I'll show you how, okay? So number one, let me check if is there a summary for this before I keep on yapping. Oh, there's no summary, okay? But it's being recorded, so don't worry, you can watch it later. All right, here we go. Quick one. All right, first of all, number one, place 5 centimeters cubed of lipase enzyme into a test tube and label it L. L for lipase, okay? So that's one, lipase enzyme. Number two, place milk. Now, why milk? Why are we talking about milk here? Because remember, milk, what is milk? Milk is fat. Milk contains fat. It's fat and water, basically, milk, yeah? So place another test tube containing milk. So you have lipase enzyme and you have lipid from the milk, okay? Then, in the milk, we're going to put 5 drops of an indicator called phenolphthalein, yeah? Now, remember, phenolphthalein is what in alkaline? Pink. So when you put that, it's going to... you also add something called sodium carbonate. So in here we're adding indicator, yeah, indicator phenolphthalein, but it won't change color unless it's alkaline, right? So we also add sodium carbonate in here as well, just to make it go pink. There you go. Okay, everyone? Good. So this is our starting reaction. All right, next, we're going to put both test tubes in a water bath of the temperature that we're testing. So if we're testing the speed of the reaction at 30 degrees, the water bath is going to be 30 degrees Celsius. Okay, we put it into the water bath. Once they both hit 30 degrees, then we start the reaction. Okay, so make sure in your practical step, you say use a water bath and measure both temperatures before you start the practical. All right, then we're going to take 1 centimeters cube of lipase from here, and we're going to put it into the test tube that contains milk. Okay, now, so that's going to go into here. Now, what do you think is going to happen, guys? Let's think about it. Well, what we're doing, we're putting the enzyme that breaks down fats into the beaker that contains fats. So the lipid that's inside this beaker is going to break down into what? Glycerol and fatty acid. So we're making lots of fatty acids now inside this milk beaker, right? So if fatty acids start to get produced, the pH of this test tube is going to go down. And how do we know the pH is going to change? Because of the indicator, which is going to go from pink to colorless. Okay, so measure using a stopwatch how long it takes for the color to disappear. Okay, and obviously it goes white. Why does it go white? Because milk is white, right? And our indicator goes colorless, but because the background is white, it looks white. Yeah, and then all you got to do is you just have to repeat with different temperatures and calculate a mean. There you go. That's it. Okay. By the way, guys, little side tip. Yeah. Whenever you do an experiment, yeah, like this, always mention repeat, get a mean. Those are always worth marks. Okay. So repeat the same temperature and get a mean. Done. All right. Good stuff. That was the decay practical. I hope that helped you. Now we're going to move on to another topic. I lost my track of thought. What topic was next, guys? Someone said protein synthesis. What, you want to do that one, yeah? Protein synthesis. Okay. Oh, come on. Where's the summary? Is there a summary? Oh man. Oh yes. Oh, there's a summary for protein synthesis. There's a summary. Fine. I'm gonna run through it, then I'll give you the summary. Okay, everyone. Here we go. DNA and protein synthesis. Okay, so basically, look. Wait. Let me just say something to you. Okay, your cell is a factory that makes proteins. Do you understand that? Your cell is a protein factory. Okay, it makes proteins using instructions that is in the DNA. Okay, the problem is the instructions are in the nucleus, in the DNA, but the machine that makes the proteins, that assembles it, is outside over here, called the ribosome. Do you understand that? So the instructions are in the DNA, which is very large, and that contains the instructions to make all the proteins. Yeah, the machine that assembles the protein is outside in the cytoplasm, called the ribosome. Okay, so step one, DNA is too large to leave the nucleus. Okay, and secondly, we don't want the whole DNA out. We just want the gene that we're trying to make. Yeah, we just want the gene. The gene is the part that. So step two, the gene is copied to produce something called mRNA, which stands for messenger RNA. So the gene is that section of that gene gets converted into something called mRNA. Okay, side point, mRNA is only single-stranded, whereas DNA is double-stranded. Okay, so mRNA single-stranded. All right, now we've got this short mRNA, which is a copy of one gene, single-stranded. It's small enough to leave the nucleus. Okay, this process where we made mRNA is called transcription. Good. So transcription is when the gene gets copied and turned into mRNA. Done. All right, and that happens in the nucleus. Okay, now the mRNA leaves the nucleus through the holes called nuclear pores. It leaves the nucleus and it binds to the ribosome. So mRNA travels to the ribosome in the cytoplasm and it binds to it. Okay, just like that. Once it binds to the ribosome, now the ribosome is going to read the instructions of the mRNA. It's going to read it. And as it's reading it, it will begin to assemble the protein. Now, who remembers what are proteins made of again? They're made up of what? Amino acids, right? So as the ribosome is reading the mRNA, it's going to assemble the correct amino acids in the correct order to make the protein, okay? So now here's the thing. Amino acids themselves cannot travel to the ribosome. They don't have feet. They can't walk to the ribosome. So there's another helper that brings the correct amino acids down. That helper is called tRNA. So this little guy over here, tRNA, helps to bring the correct amino acids to the ribosome, okay? Now, what's special about tRNA? It looks like a fork with 3 lines, okay? Those 3 lines are called anticodon, okay? So if you look here, there's 3 lines on the mRNA called codon or triplet code. So the mRNA's got 3 lines, and the tRNA's got the opposite 3 codons, so it will join together. Okay, tRNA, which has the complementary anticodon, will bring the correct amino acid. And then it will just assemble it, bam, 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 bam. All the amino acids get assembled, and you have a chain of amino acids, and that is called a protein. And this process, where the ribosome assembles the amino acids, this process is called translation. There you go. And if that's a lot, here you go, everyone. Here's your nice little summary of protein synthesis. Okay, that all these points explain from the start to the end how a protein is formed. Nice. Okay, so screenshot that one as we're about to move on to the next topic. Okay, let me see what else. What's another hard topic that we haven't done yet? So we've done that, we've done this. Oops. Say that again? Oh, Humayma asked if I'm tired. No, Humayma, I'm actually just starting. I'm about to start teaching you properly in a second. Biology. So we've done this, we've done... Guys, I think the main topics are covered, can't lie. Okay, okay. Very quickly, speciation. We never done speciation. Let's do that. If you guys are tired, then of course I do understand. And I will, after this, we'll finish, okay? Here we go. All right, everyone. Here's our last topic before we... someone's saying microprotein. Someone's saying contraception. I don't know if you want me... hold on a minute. Microprotein, yeah? Let me just check my predicted topic list if that's coming up. Maybe. Okay, let's do speciation and we'll see, yeah? Let me see. Let me call AQA and ask them if microprotein is coming up. All right, speciation. So, what is a species? Species are individuals that can reproduce to create fertile offspring, yeah? So, for example, if I take a male horse and a female horse and they make a baby horse, if this baby horse can make more babies, that means that these guys were the same species, okay? But if the baby horse cannot make more babies, then that means they were not the same. For example, you could take a male horse and a donkey and the female donkey, okay? And you can make them have a baby called a mule, yeah? The problem is the mule cannot have any more children, okay? So that means the male horse and the donkey are not the same species, yeah? All right. So I don't know what I'm laughing about. So mule is infertile, okay? So how does a species form? This could be a nice little question, yeah? Nice little question. All right, here we go. So how does a new species form? So first of all, we have can you see over here on this island, we have these black dots. So these are all the same species. They're all, let's say for example, they're all like monkeys, yeah? They're all the same monkey. But there are 2 sets of these monkeys, one on this side of the island and one on the other side of the island, okay? They get separated by something called a geographical barrier. So it could be like a mountain range, it could be something just basically separated them and isolated them. So monkeys from here cannot go there and monkeys from there cannot go there because they're separated, okay? Now, each side is going to have natural different alleles, okay? So there are each population has different alleles. So maybe over here, the alleles on this side are different to the alleles on this side, okay? So we can say they both have variation because of what? Mutation, okay? Secondly, the conditions on each side is also different. Maybe over here it's very, very hot and sunny and bright, and over here it's very dark, rainy and wet, okay? So what does that tell us again? It tells us that the selective pressure is different on each side, Selective pressures are different on each side. So that means that natural selection happens on that side and on that side, but the advantageous characteristics are different. Maybe over here, having less fur is advantageous, but over here, having more fur is advantageous. Do you get it? So both sides will have natural selection, but they will have natural selection in different directions. So natural selection happens in both populations, but different advantageous genotypes are on each side of the barrier, okay? So what happens is, over time, they become so different that they cannot interbreed successfully. So after, like, let's say 100 generations later, you bring this monkey here and bring one monkey here, and you try to make them breed, but they become so different that they can't breed anymore. So if they can't breed anymore, what does that mean? That means that they are not the same species. Okay, this is now a completely different species to that one, and that is speciation. Okay, so speciation is when a new species forms. Before they were the same species, they could breed, but now they're completely different. Okay, that's how it works. All right, guys, there you go. So that's fine, done. Okay, so blah, blah, we have time. We'll go till 10:30. Okay, and after 10:30, I'll let you guys go to sleep. DNA some. Let's see, what else have we done? We've done this. Okay, let's go through. We have done the menstrual cycle. Wait, this is one. What is this? Biotech. All right, one sec. Okay, we'll go through fossils. Yeah, this is an important one. We'll do fossils, and then we will. Here we go, guys. Let's quickly go through fossils. Yeah, here we go. All right, what is a fossil? Fossils are the remains of dead organisms. Yeah, easy beginning to the topic. Now, they can form in three ways. Number one, by gradual replacement. Number two, by casts and impressions. And number three, by preservations. Okay, those are three ways that we can form remains of dead organisms, aka fossils. All right, so what is gradual replacement? Gradual replacement is when the animal dies and gets buried in sand. Okay, then over the soft parts get decayed and eaten by microbes. So like the meat and flesh and skin, they get decayed, but the bones remain. Yeah, now the bones don't get eaten by microbes. So what happens is, over several years, the bones get replaced by stone. They get turned into, they get replaced by minerals, which makes them tough like stone. Okay, and then we discover it. Archaeologists discover it, and that's where you get fossils. So that's one type of fossil. Okay, number two, casts and impressions. So if an organism is buried in something soft like clay, okay, or they step on something soft like clay, then over time that clay hardens around the organism or around the imprint, and that forms a cast. Easy. And then from that you can see either the whole organism, or you can see bits of the organism. Okay, and my favorite, preservation. So preservation is, there are three types of preservation. Okay, let's start with the first one. If an animal dies in a material called amber, amber is a yellow stone that has no oxygen inside it. So if an animal like a spider, for example, falls into a pit of amber, okay, and the amber hardens around it, there is no oxygen in amber. What does that mean? There is no decomposers, okay? So decomposers cannot do respiration. There's no oxygen. So what happens? The organism that's inside the amber will not get broken down. So it will remain preserved forever, okay? Now, another one is if an animal gets frozen in glaciers, okay? Here, there is oxygen, but it's so cold, yeah? It's so cold that the decomposers enzymes do not work. So this time, there is decomposer, there is oxygen, but it's too cold for the enzymes to actually break it down. Okay, and the last one is in peat bogs. Peat bogs are acidic, or areas that are like swampy, muddy areas, and they are very acidic. Okay, so they're too acidic. Again, if an animal dies in a peat bog, because it's so acidic, the decomposers cannot break down the animal because the enzymes just get damaged. So again, the animal that died in the peat bog gets preserved forever. Okay, good. So those are the three ways that we did fossilization. Okay, so guys, it's becoming 10:30, so it's getting quite late. there was some people saying plant hormone. Yeah, oh, okay, fine. Let's do plant. Let's do plant hormone. Okay, let's do plant hormone. Come on, let's do it. Lock in, everyone. All right, plant hormone time. Let's go. All right, we'll finish after plant hormones. Yeah, we'll finish after plant hormones. Okay, fine. You ready, everyone? Let's go. All right, plant hormones. So, auxins are an example of plant hormones, okay? And they are found at the tips of the shoots and the tips of the roots, okay? They're found at the shoots and the roots, two different places, yeah? Now, those auxins, they do different things in the shoot and the root. They act differently, okay? They move differently. So, let's start with what they do. Auxins control cell elongation. So they control how long a cell gets. They do not control cell division, okay? They don't control cell division. All right, so let's talk about this, yeah? So what's the point of auxins anyway? The point of auxins is it allows the plant to respond to light and gravity, yeah? So without auxins, the plant cannot respond to light and it cannot respond to gravity. So let's talk about it. Now, at the shoots are the part at the top of the root, okay, of the plant. The shoots over there, auxins cause the cells to get longer. So more auxin, cell gets longer. But in the roots, auxins don't do that. They do the opposite. In the roots, they stop their cells from getting longer. And everyone confuses this, so make sure you don't confuse it, yeah? Here's a line. At the shoots, longer. At the roots, stop longer, yeah? Okay. So let's look at the shoots and how they respond to light. Okay, so look, let's zoom into the shoot over here. All right, so we're going to zoom into the tip of the shoot. Okay, now here we have initially auxins are evenly distributed at the tip. So can you see how at the tip of this shoot the auxins are initially evenly distributed across the tip? Yeah. Now, when the sun shines, so the sun is shining on the tip, yeah, one side becomes shaded. Auxins move to the shaded side of the tip, okay? And the bright side has no auxins. Okay, everyone? Now, next part. Remember how we said earlier that in the shoots, the more the auxins, the faster it grows and gets elongated longer. So this side of the tip gets longer, okay? You guys understand that? So the tip is not going to grow evenly upwards. One side of the tip is going to grow faster than the other side. So it's not going to go upwards. It's going to grow what? Which way? It's going to grow towards the sun. Okay, everyone? So the tip grows towards the sun. And Dishan, can I have some water, please, bro? Okay, it grows towards the sun. Does that make sense, everyone? And this is called phototropism. A bit of water, please. Okay, so this is called positive phototropism. All right, thanks. Okay, moving on. So next part is, let's look at the roots. Okay, the roots. Let's have a look at. Here's the root. Okay, here's the tip of the root. Now, again, we're gonna start with the auxins being evenly distributed on the tip. Yeah, but this time gravity is gonna pull the auxins downwards. Okay, so look, can you see how the gravity is pulled the auxins to the lower side of the tip? Yeah. Okay. Now, remember, in the roots, the more auxin, the slower it grows. Okay, so this time the lower side is not going to grow faster; it's going to grow slower because this is the root. Remember, auxins act differently in the root. Yeah. All right. Now, that means the top side is going to grow faster. The top side is going to grow faster than the lower side. Yeah, and as a result, the root is going to grow downwards towards the earth, okay? And this is called positive gravitropism. Good stuff. All right. Now, here's the question. We have a root. This is a shoot, okay? So this, it says root here. Oh, 1 second, hold on. Okay. This should say shoot, yeah? I know it says root, but it should say shoot. So here we have a shoot. Right now it's horizontally, okay? It's horizontal. We know that the auxins are, because it's horizontal, the auxins are going to be pulled down because of gravity to the lower side. So, will the lower side grow faster or slower? It's a shoot. Will it grow faster or slower? Well, because it's a shoot, we know that the more auxins there are, the faster it will grow. So as a result, it's going to grow upwards towards the light. Okay, everyone? All right, finishing off plant hormones. So we also have, okay, ignore this, ignore this, ignore this, ignore this, ignore this. Forget that. All right, so commercial plant hormones, okay? So we have auxins can also be used commercially. So in fact, in like big plant factories for what? For killing weeds, okay? So what is a weed? A weed is an unwanted plant, and usually weeds have broad leaves, okay? They have broad, I can't even spell, sorry, my bad, guys. They have broad leaves, yeah? So the auxins, they kill the weeds which have broad leaves. All right, and this is known as selective weed killer. So they kill the broad leaves and they don't kill the crops, which are the good plants that have narrow leaves, okay? So they're called selective weed killers. All right, number 2. Sometimes when you make a plant cutting, okay, so here's a plant and you want to copy this plant, you want to make a clone of it. What do? We cut a little bit of the plant and then we put it in something called rooting powder. Rooting powder also contains auxins. What does it do? Well, when you put it into rooting powder and you take that little piece, now that will stimulate the cutting to grow roots rapidly. So if you put that little cutting into the soil, it will grow roots and then quickly turn into a plant, okay? So the last bit. So let me just quickly go back. Auxins, they help plants to respond to light and gravity. Number 2, they can be used in selective weed killer. And number 3, they can be used in rooting powder to help cuttings turn into grow roots. Okay? All right. What's the next part? Okay, ignore this. Now let's look at gibberellins. Yeah? What do gibberellins do? Gibberellins control dormancy. So sometimes a seed will not germinate until the conditions are right. So they stay in the soil. They're not going to grow because they're probably waiting for the sun to come and stuff like that, yeah? But we don't have time for that. We want them to germinate straight away, yeah? So we use a hormone called gibberellins, and they force the seed to germinate, okay? They force the seed to produce roots and shoots and to grow. So that way we can grow plants all year round, no matter what the conditions are outside, we can keep on growing plants in our plant factory, okay? Also, gibberellins can also induce flowering, so it can force the plant to grow flowers, okay? And also, they can force the plant to grow larger fruits, okay? So 3 things. 1, what was it? Controlling dormancy. Number 2, making flowers. And number 3, to grow larger fruit. And the last one is ethene, okay? What does ethene do? Ethene is used in fruit ripening. So when fruit is ripe, it's very soft. So if you're a farmer and you want to transport strawberries, you don't want to make them ripe and then transport them because they will get damaged because they're too soft. Okay. So what we do is we wait. We pick them a bit earlier when they're unripe and they're hard. Okay. And on the way to the destination, we spray them with ethene, so that way they can ripen on the way. So when they get delivered, they don't get damaged, okay? And that's ethene. And we shall conclude it on the party time. So tomorrow is your big biology party, everyone. Hopefully you shall. Let me vote for this one. I want to pick Zishan. Okay, so hopefully you guys smash your biology paper tomorrow. Let me know how it goes afterwards. And then guess what? Chemistry and then physics. Oh, and then I'm done. Science is done.
You're happy.
I'm going to be very sad. I'm going to miss you guys. All your funny comments in the chat. But hopefully you smash it and then come back and let me know how it was, yeah? And yeah, Nishan, any goodbye song for them? Goodbye lullaby?
Nah. Not today.
Alright then. We're going to end it nicely.
No.
Safe, everyone.
Thanks so much, guys.
TYSM.
Moderator has just put the final code as well, just as a reminder. We got this for you guys on this website, so go there, use it while stocks last. It might expire after tonight, so the live stream's done now. You know, if you ask your parents, your caretaker, whatever, to get the package, you might think, what's the point? You've only got a few exams left. You only have a few exams left, but you want to leave your exams on a high. Yeah. Not a high as in that high, but you want to leave on a grade high. Yeah, so buy the package, it becomes only £19. Use that live stream 19, lose that live stream 40, sorry, and you comes.
Oh,
£19 only.
do make dua for us. Someone said they make. Yeah, bro,
Dua,
make
yes.
that nice dua. Yeah.
Yeah.
All right then.
Okay, guys.
Safe.
This is the Buckme and Westfield. Sorry.
I'm just gonna read some of their comments. I actually never get
This
to read
person's
it.
an advocate somehow.
Nah.
Moderator.
Oh, let me see what. Okay, write something mad in the comments. Write the maddest thing you can write.
See
Let
if
me
you can bait Mohammed. See if you can bait Mohammed somehow. Guys. Can you bait Mohammed?
I see the edge of mosque.
Who's that?
How's that baiting me?
Who can bait Mohammed?
What does bok mean? What does
That
bok?
means like meet me. That might mean the alleyway. Meet me in the alleyway.
Oh, bok mean the alleyway.
What's that one?
Buckminster Florins.
What's that?
Mike, what? Mike Oxlong. Who's that? Mike Oxlong. Oh! Oi! That you admins are mad. Masturbator. Oh! You guys are mad. Wow, you guys are actually so mad. You guys are mad. Wow. I'm reading your. This is the first time I'm actually reading your comments live on chat.
How they are?
Well, this one, the dog bid is saying, "Will you marry me?" Raw.
I don't even take away this guy's mod. The one, the guy
Delete.
that's saying buck me.
Delete buck
Yeah,
me.
get rid
Wait,
of his mod.
are you making like random people mods?
Not random, like if they say something funny, we just give them mod.
Oh no way.
Yeah.
Oh.
Did you get rid of his mod?
This one looks like a genetic mutation.
Huh? What they say?
This one looks like a genetic mutation.
What? Who's that? Ishan.
Yeah, get him.
Give him a mod.
Block him from the next live
Nah,
stream.
give him the mod. Give A Ishan K9Y. Give him the mod. Decent one.
Oh, that's quite funny, guys. This one kind of take all your bait. Muhammad looks like a Roblox character. What the hell? Oh. Nah, that's mad. That's mad.
Oh dear.
Should I sleep more or wake up early? Wake up early. Oil up. Ah, this guy must be some.
Oil up, he's oil up.
That's an old thing, mate. Oil up. That's not this year. 7 year. Can I drop a subject the night before? Nah, mate. You've been doing it. You've been doing it for 5 years. Why would you drop it the night before? Just go and get a U at least. Bro, this guy's crazy. This guy's a bit angry. Why should chef us up? My name is you.
The thing is, they're trying too hard now.
Yeah,
That
yeah.
is funny. Anyway.
But
You know when you tell them twice. Okay.
or are they trying? Are they just doing like
What's
like?
this one?
Huh?
My mate has what?
My mate has huge as. He
What does
can
that
carry
mean?
us to 9s.
What's that?
I don't know. I don't put huge has. Oh, this is better than the guys. Why are they using what they learned as a diss to diss us? How do you rap to eat? Bro, these guys are mad. Listen, let me tap. I'll put my adult cell clone in. Bro, these guys are mad. These are- I'm gonna delete the live chat from the next ones.
Whoever's getting too excited, get rid of their mod.
I'll sacrifice my life for. Oh, okay, you guys are. Can't lie, they're not that funny. They're not that funny. You guys are not funny.
Okay, guys.
Not-
Take care. And maths paper 3 live on Tuesday night. Subscribe, get us to 100k, like the vid, do all of that stuff, and we'll see you guys on the next one.
Yeah, see you guys.